Yes, Dr. Pradeep.
Good morning to all. I extend a warm and heartfelt welcome to each and every one of you for today's enlightening webinar. I am Dr. Pradeep Golani, the co-convener of this event, and it fills me with immense pleasure and joy to witness such a diverse and engaged audience gathered virtually. Your presence here not only adds to the vibrancy of our discussions, but also underscores our collective commitment to advancing knowledge in our field. I am genuinely anxious about the prospect of delving into the insightful and collaborative conversations as we embark on this intellectual journey together. By consistently providing captivating webinars, SPSR has established itself as a premier platform for knowledge exchange and professional development in the pharmaceutical sciences. I am confident that today's session will uphold this reputation, with attendees benefiting greatly from the expertise shared by our esteemed speakers. Let us seize this opportunity to learn, engage, and contribute to the advancement of our field. Welcome once again, and I look forward to a rewarding webinar experience for all. Thank you for joining us today. Today is a significant milestone as we gather for the 48th SPSR International Webinar on drug repurposing, exploring new therapeutic avenues for existing drugs. This event is a part of a distinguished series hosted by the Society of Pharmaceutical Sciences and Research under the esteemed mentorship of Dr. Shashi Alok, National President of SPSR. Under his guidance, the SPSR continues to play a pivotal role in advancing knowledge and innovation in the field of pharmaceutical sciences. The SPSR is dedicated to advancing the pharmacy profession through a multifaceted approach. Our objectives encompass the organization of conferences to campaign the causes of pharmacy, contributing to programs and services that address the health needs of India and focus on disease prevention. Additionally, we actively recognize and honor distinguished pharmacists who contribute significantly to the encouragement of pharmacy profession. The core of our mission is the promotion, facilitation, and the acquisition, dissemination, and exchange of pharmacy knowledge, ensuring a robust flow of information about the practice of pharmacy and pharmaceutical services. Through these objectives, we aim to elevate the standards and impact of pharmacy profession, fostering a dynamic and collaborative environment. We are truly honored to welcome our esteemed speaker, Professor Dr. Ravichandran, Professor and Head, Pharmaceutical Chemistry Unit, Faculty of Pharmacy, AIMST University, Semeling, Malaysia. Professor Dr. Ravichandran brings invaluable expertise to our event, and we eagerly anticipate gaining insights from his wealth of knowledge in the field. His presence enriches our discussions and adds significant value to our exploration of the latest advancements in pharmaceutical services sciences. We are grateful for his participation and look forward to a truly enlightening session. In our esteemed gathering, we are honored to have the distinguished presence of Mrs. Monika Sabharwal, the convener of this webinar, National Secretary, Society of Pharmaceutical Sciences and Research, and Managing Editor of IJPSR and IJP. Her multifaceted contributions and leadership in these capacities underscores her commitment to advancing the field of pharmaceutical sciences and research. We are privileged to have Mrs. Monika Sabharwal with us today, guiding our webinar with her expertise and dedication. Without further delay, I have the distinct pleasure of inviting Mrs. Monika Sabharwal to extend a warm welcome to our esteemed guest speaker, Professor Ravichandran.
Thank you so much, Dr. Pradeep Golani. Hello, everyone. It is with a great pleasure and honor that I, Monika Sabharwal, stand before you as a convener of 48th SPSR International Webinar on drug repurposing, exploring new therapeutic avenues for existing drugs. Today marks yet another milestone in our relentless pursuit of knowledge and innovation in the field of pharmaceutical sciences. Organized by esteemed Society of Pharmaceutical Sciences and Research SPSR, this webinar serves as a backend of collaboration and exploration, bringing together experts from around the globe and delve into the exciting realm of drug repurposing. I am pleased to announce that we have privilege of hosting Professor V. Ravichandran as Professor and Head of Pharmaceutical Chemistry Unit at Faculty of Pharmacy, AIMST University, Semeling, Malaysia, as our distinguished speaker for this event. Professor Ravichandran's expertise and insights are sure to shed light on the immense potential of repurposing existing drugs for novel therapeutic purposes, offering perspectives and groundbreaking research findings to our audience. I'm happy to share that we have received overwhelming response with more than 3,000 registrations pouring in from 20 countries worldwide. This global participation is a testament to the universal significance and impact of the approach, the landmark of our 50th SPSR webinar series. We reflect on tremendous support and enthusiasm received from our global community. Each webinar has been testament to our commitment to the excellence and the advancement of pharmaceutical research and education. The positive feedback we continually receive as a testament to value and impact of our initiatives, further fueling our passion and dedication to uplift the pharmacy profession and foster excellence in the pharmaceutical sciences. It is indeed an immense honor for extend a warm welcome to Dr. Shashi Yalu, the esteemed National President of SPSR. His visionary guidance has been instrumental in orchestrating a remarkable transformation within SPSR, positioning it as a beacon of excellence in the field of pharmaceutical sciences. I would also like to express my sincere appreciation for the contribution of Dr. Pradeep Golani, the co-convener of this webinar, and the organizing committee members, Dr. Sanjay Nagdev, Dr. Payal Mittal, Dr. Saurabh Kose, Mr. Ankur Agarwal, and Mr. Junaid Tantre for their tireless efforts, which have played a crucial role in transforming this event into a reality. Behind the scenes, there's an exception team who is always a backbone for a seamless organization. Their tireless effort and dedication have made this webinar possible. I extend my sincere gratitude for each and every SPSR team member for their thorough attention to the detail and their passion to deliver excellence, which have made this webinar possible. I extend my heartfelt gratitude to Professor V. Ravichandran for his valuable contribution to the webinar and all of you for your continued support and participation. Together, let us continue to strive for excellence and innovation in the pharmaceutical sciences. I extend my deepest gratitude to each one of you for your enthusiastic participation and valuable contribution to this webinar. Your engagement fuels our pursuit of excellence in the field of pharmaceutical sciences. I am confident that today's discussion will be both enlightening and inspiring for us. Once again, I extend my sincere gratitude to each one of you for integral part of 48th SPSR International webinar on drug repurposing, exploring the new therapeutic avenues for existing drugs. Your active participation and engagement are invaluable contribution to our pursuit of knowledge and the progress of pharmaceutical sciences. Together, let us seize the opportunity to learn, collaborate, and innovate together. Let us harness the power of drug repurposing to explore the new therapeutic avenues and address unmet medical needs, ultimately improving the patient outcomes and advancing the healthcare globally. Stay at forefront of innovation with the SPSR. By subscribing to our YouTube channel, you gain exclusive access to upcoming webinars, experts' insights, and groundbreaking research in the field of pharmaceutical sciences. Join our vibrant community of knowledge seekers and actively engage in a thought provoking discussion that will shape the future of the healthcare. Do not miss the latest updates and remember to subscribe our YouTube channel and hit the notification bell icons for the instant alerts. Join us on discovery and innovation as we work towards the advancing of pharmaceutical sciences for the betterment of our society. Without further ado, let us begin with the 48th SPSR International webinar on drug repurposing and exploring the new therapeutic avenues for the existing drugs. Over to you, Dr. Pradeep Golani.
Thank you, Mrs. Monika Sabharwal, for allowing me to conduct this webinar. And now let's begin with the brief introduction to guest profile. Dr. V. Ravichandran is a distinguished figure in the field of pharmaceutical chemistry, holding the position of Professor and Head of Pharmaceutical Chemistry Unit at the Faculty of Pharmacy, AIMST University in Semeling, Malaysia. With a robust educational background and extensive professional experience spanning for over 2 decades, Dr. Ravichandran has made significant contribution to academia, research, and scientific community engagement. His educational journey commenced with a bachelor's degree in pharmacy from Dr. MGR Medical University, Tamil Nadu, India, followed by the master's degree M.Pharm in pharmaceutical chemistry and PhD in pharmaceutical sciences from Dr. H.S. Gaur University, Sagar, India. Throughout his academic pursuit, he has received prestigious fellowships and grants, including Junior Research Fellowship funded by the University Grant Commission and Senior Research Fellowship funded by the All India Council of Technical Education, demonstrating his commitment to scholarly pursuits. Dr. Ravichandran's professional trajectory is marked by various academic appointments and leadership roles. He has served as a faculty member at esteemed institutions such as Al-Shifa College of Pharmacy in India and KMCH College of Pharmacy, where he contributed to both teaching and research. His tenure at AIMST University, Malaysia, has been particularly noteworthy, where he has progressed from assistant professor to his current position as professor and head of Pharmaceutical Chemistry Unit. In addition to his teaching responsibilities, Dr. Ravichandran has demonstrated a prolific research record focusing on diverse areas such as medicinal chemistry, molecular modeling, phytochemical analysis, and analytical methods of development. His expertise also extends to green synthesis and characterization of metal nanoparticles, reflecting his commitment to sustainable practices in pharmaceutical research. Dr. Ravichandran's research endeavors have been supported by numerous grants from reputable funding agencies such as the Ministry of Higher Education in Malaysia, underscoring the significance and impact of his work. His scholarly output is extensive, encompassing publications in both national and international journals, along with contributions to book chapters and scientific presentations. Beyond his academic and research accomplishments, Dr. Ravichandran is actively engaged in external academic activities, including serving as the adjunctor for doctoral and master's thesis, participating in examination moderation panels, and delivering invited talks and guest lectures at various institutions globally. His contributions have been recognized through awards and accolades, including the PM SDS Distinguished Pharmaceutical Scientist Award, Dr. APJ Abdul Kalam Award for Teaching Excellence and Scientific Excellence, and the International Scientist Award. Dr. Ravichandran's multidimensional expertise, coupled with his dedication to advancing pharmaceutical sciences, makes him a prominent figure in the academic and scientific community, both in Malaysia and internationally. His leadership, research progress, and commitment to education continue to inspire students, researchers, and fellow academicians worldwide. It brings me immense joy and pride to share that we have garnered the participation of over 3,000 registered attendees from across the globe, representing a diverse array of countries. Among them, we have significant presence from India, Algeria, Azerbaijan, Bhutan, Bulgaria, Ethiopia, Germany, Ghana, Indonesia, Ireland, Kenya, Kazakhstan, Malaysia, Mexico, Mongolia, Nepal, Nigeria, Portugal, Pakistan, Philippines, Poland, Saudi Arabia, Thailand, and UAE. The overwhelming response from such a wide-reaching and varied audience is truly humbling. We extend our heartfelt gratitude to each and every participant for choosing to be a part of this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Ravichandran, with us today. Welcome, sir. Kindly, I request you to share your PowerPoint presentation and talk.
Okay, thank you, Dr. Pradeep. First of all, I would like to thank the Society of Pharmaceutical Sciences and Research for inviting me to deliver a talk in this 48th webinar. Particularly, my special thanks to Dr. Sasi Aluk for conducting me to deliver the webinar today on drug repurposing. And I take this opportunity to thank Dr. Pradeep and Dr. Monika, those who are acting as convener today. And my special thanks to the participants. I think just now Dr. Pradeep mentioned more than 3,000 participants. So my special thanks to everyone. So with your permission, I share my slide. So my slides are visible, sir?
Right, sir. It's visible, sir.
Okay, thank you, sir. So today we are going to see drug repurposing. So from the audience, the distribution, the registered audience, I came to know that more than 50% are the students and remaining 50% contains researchers, scholars, scientists, and professors, lecturers. So sometimes I will move to, means I have to cover some basic because more than 50% of the participants are students. So I request the lecturers, professors, and scientists, please bear with me when I'm going first little bit the basic things. So drug repurposing, so exploring new therapeutic avenues for existing drugs. So this one line will explain what I'm going to talk today, the drug repurposing. Yes, when you want to find out the new drug, so as we all aware about that, the new drug invention is not that much easy task. So it is a complex process. It will take minimum 10 to 17 years to bring a drug into the market. Then it has to cross many stages and we have to spend a lot. So it's not cost effective. Then we have to think about the laborious and the technologist and the other things like time. The most important thing is time. So all these things made the scientists to think about drug repurposing. So before going to start my talk, 1 minute. So this is my disclaimer. So my slides, whatever I am giving in my PPT, it's not my own. I have taken the pictures and the tables and other things from different data sources available in the web, articles, lectures, and tutorials, and few things I have taken from other researchers' PPT also. So I don't have any rights on that. So all the rights, the authors are on their copyright. So it's not, I'm not related with that one. So I have taken from the sources. Then whatever I'm going to discuss for the another 1 or 1.5 hours, it's my personal opinion, suggestion, and whatever I'm going to comment, it is everything is my personal capacity only. It's not related to anybody else or to any of the institute, including my institute. So everything is my personal opinion and suggestion. So I can start my talk with the golden words by the Nobel laureate, Sir James Clark, who is the Nobel Prize winner of 1988 in physiology and medicine. So what he told the most fruitful basis for the discovery of new drug is to start with an old drug. So this is the concept what we are going to discuss today, that is drug repurposing. So whatever the things we want to find out the new drug, we have to start with the old drug. So what is the benefit? Why we have to go for this drug repurposing? What is the importance? So that only we are going to discuss today. So in old drug, there are two ways are there. We can find out the new uses of old drug, what we are going to see today, that is drug repurposing. We are going to find out the new therapeutic uses of the old drug, means which is approved, already approved by FDA, which is intended for some other use, some different use, but we are going to find out and use for new therapeutic. Disease means new therapeutic for any disease or disorders. That is new uses of old drug. Otherwise, the other thing is new lead from old drugs. This is the other techniques, which is both are going to use the old drug for the new drug. But the first one is the old drug. We are not going to disturb the structure of the old drug. Either we can increase the dose or change the formulation or find out the new target for the old drug. That is new uses of old drugs. So that is also called as drug repurposing. The second concept is we are not going to use the old drug, but we can take that one as a lead drug. By doing some bioassays, we can find out that drug is having some activity on any new target. Then we can take that molecule as your lead and we can optimize it to bring us your drug molecule. So in drug discovery, you all are aware about there are three things are there in chemistry: hit molecule, lead molecule, and drug molecule. So here we are going to take the lead molecule as the old drug as a lead molecule. So after that, we have to do some modification in the structure. We can change the functional group, we can change the something in the basic nucleus, or we have to do some changes in the molecule and increase the potency of that particular drug, or not increase the potency, increase the efficacy against the new target. Because some drugs you can take, it's having main therapeutic effect, but it's having lot of side effect. So we can minimize the therapeutic effect and bring any one of the side effect as your main therapeutic effect. That is called as the selective optimization of side activities. That is the second process. New lead from old drugs means we can bring the side effect as your main therapeutic effect and reduce the minimize the main therapeutic effect of that drug. That is called a SOSA, selective optimization of side activities. So this one is drug repurposing. So for the drug discovery, so in drug discovery, so just now I told it is not a simple process. It is a complex and expansive process from the identification of the disease, identification of target. Then we have to validate the target. Then you have to find out the drug molecule, the lead hit molecule for the target. Then hit molecule will be optimized to lead molecule. Then the lead molecule will be optimized to drug molecule. Then you have to file investigational, you have to go for formulation, then preclinical studies, then file investigation, new drug, then new drug application. Lot of things are there to bring out one drug into the market. So it is a complex and expensive. The more than that, it is a time-consuming process in conventional drug design. It will take near about 10 to 17 years to bring a drug into the market. Not only this, the other important thing is the failure rate, because the drugs are designed and preclinically it's showing excellent effect, but when it's coming to clinical, there is a lot of failures because of its toxicity. 90 to 90% of the drugs are failed in the clinical trials. So that is the main drawback when you are going for new medical entities or new chemical entities. That is the main drawback. So only the scientists now, they are moving towards the drug repurposing. And more than that, when you are developing the new drug in drug discovery, there are a lot of controlling body and the regulations are there. For example, when you are doing the laboratory work, then you have to follow the good laboratory practice, good in the good clinical practice, good manufacturing practice and marketing act. Then Health Insurance Portability and Accountability Act. There are many acts and rules and regulations are there to follow. So we have to spend a lot to bring a drug into the market. Near about, I think 0.8 to $1 billion they are spending to bring a drug into the market. So cost-wise, it is highly expensive, the conventional drug. So these are the different stages when we are doing the conventional or rational drug design approach. So conventional drug design approach, it is a trial and error method. But in rational drug design approach, we are using, first we are studying about the target. So what is the target which is responsible, or which is the causating factor for the disease, and what are the receptor or enzymes are going to be affected by the disease. First, you study thoroughly about the disease and understand what is the target. Then you have to design the molecule by using the sophisticated techniques. There are a lot of techniques out there. By using that, we are going to find out the drug. That is the rational drug design approach. But in earlier, the traditional approach or conventional approach, once the disease was identified, what they did, they developed the bioassay and screened whatever the compounds they were having, screened against the particular disease without knowing anything about the target. So just they want to find out the drug for the particular disease. That was the conventional drug design approach. So here, mainly this is indicating the rational drug design approach, the particular slide. So once the disease is identified, then you have to understand about the target. What is the protein or receptor involved in that particular disease? So you have to isolate the proteins. Then you have to study about the proteins and try to understand what is the structure and function of the proteins by using any one of these omics studies, either genomics, proteomics, or biopharmaceutics. Then once it is ready, then we can go for high throughput screening. High throughput screening is your wet lab technique where we are having the synthesized compounds. So you have to develop the bioassay, suitable bioassay. Then using sophisticated instrument, you have to screen the compound against the particular target. That is high throughput screening. If you are not find out any non-synthesized compounds, means here we are going to use the synthesized compounds library. Means we are going to do the practical experimental work. So if we found out that any of the hit molecule from these techniques, then we have to optimize those drugs by using the computational chemistry. If suppose we are not getting any drug from here, then what you have to do, we have to go for virtual screening. Virtual screening means here in high throughput screening, we are using the synthesized compounds and we are doing the experimental bioassay. But in virtual screening, we are going to use the virtual molecules, means the drawn molecules. It's not the synthesized molecules. So by using these techniques, we can find out from our virtual library, we can find out any of the compounds is having any affinity towards the target by using. There is a lot of techniques are there like QSAR, pharmacophore mapping, docking studies, dynamic studies, many things are there. We can use with the suitable technique to find out the hit molecules. Once here, if it is confirmed from high throughput screening, then we are having the synthesized molecule, then we can go for further studies. But here, it is not the synthesized molecule, it is your designed molecule. So once, if you find out your target, means the hit molecule from here, then we have to synthesize the molecule. For that, we cannot go for the conventional synthesis one by one. Instead of that, we can go for combinatorial chemistry. That is, combinatorial chemistry is simultaneous synthesis of at least minimum 10 compounds at a time. That is called as combinatorial chemistry. So you have to synthesize multiple number of compounds in a short period of time. Once it is synthesized, then you have to do the bioassay screening, then find out the compounds. Then we have to optimize the compounds for toxicity as well as the efficacy. Then you have to use any one of the molecular modeling technique to optimize the compound. Once it is optimized, everything is ready, then we can go for in vitro or in silico ADME models. So earlier we are doing in vitro, but nowadays there are a lot of online tools, softwares are there to do ADME absorption, distribution, metabolism, and excretion models. We can use any one of the in silico models to minimize the use of animals. Then later on, you have to go for preclinical testing. If everything is okay, then we can move to the clinical trials. So this is the now rational drug design approach techniques. So here where we are going to use this techniques. So here. So once the disease is identified, so instead of these techniques, we are going to go for the alternative approach, either the selective optimization of side activities or drug repurposing. So just now I told the selective optimization of side activity means new uses of the old drug by optimizing the side activities. But in drug repurposing, we are not going to do any optimization, just we are going to find out the new use of the old drug without any modification. That is the difference between the first concept and the second concept. So for example, new lead from older drugs, that is selective optimization of site activities. Earlier, the sulfonylamides are there. Then they found out the sulfonylamides are having antibacterial activity. Then there is a lot of compounds are developed from this sulfonylamide. There are a number of derivatives later they identified. The same thing for chlorothiazide and promethazine as antihistamine and chlorpromazine as antipsychotic drugs. So these are the some examples. Very few examples. There are many other. Very few known example I have given for the new lead from older drugs. Means we are taking the lead. Then later on we are going to do the introduce the functional group or change the functional group, doing some changes in the structure and make it as a therapeutic one against the particular disease or disorders. So that is called as new lead from older drugs. So here we are not going to use this molecule as a drug, but after doing modification, we are going to use as a drug. So only it is new lead from older drugs, that is selective optimization of site activities. Then new uses of old drugs, that is the concept of today's talk, that is drug repurposing. So before going to see what is the concept, what are the strategies, why we are going for that, and what is the advantage, what are the challenges. First, we see the fruitful output of this drug repurposing technique. One of the best example is the sildenafil. That is originally developed for the treatment of hypertension and angina pectoris. But later on, when they are doing the clinical trials, they found out it is a good drug for managing erectile dysfunction. So it has currently banked a huge market. As you all are aware that it is available in the name of Viagra. Then bupropion is an antidepressant drug. But patients taking these drugs, those who are smokers, later on they found out they helped them to give up smoking. So now it is used as anti-smoking aid, that bupropion. Then the another drug is estimazole, is a medication used in the treatment of allergy. But later on, they found out it is the best drug for anti-malarial and anti-cancer. All are it is approved drugs, FDA approved drugs for the new users. So here, sildenafil for erectile dysfunction, it is available in the market. Then bupropion, it is approved one. And estimazole also approved for anti-malarial and anti-cancer agent. So clinical effect of drug or compound. So as we all are aware about that, whatever the chemical compound you are taking or the target, either it is a receptor, protein, amino acid, whatever it is, everything is made up of functional groups, the chemical groups, chemical atoms. So whenever you are taking two molecules, whether it is a small molecules or small molecule macromolecule or micromolecule macromolecule, there will be an interaction, chemical interaction will be there. So when our drug molecule is going to interact with the receptor or protein or DNA, then it is going to do the physiological changes that is responsible for the pharmacological output or biological output. So the reaction, how the interaction will take place, either it is your van der Waals interaction or hydrogen-hydrogen interaction, means hydrogen bond formation, or hydrophobic interaction, or hydrophilic interaction. There are a lot of interactions that are based upon the functional group, what the drugs and the target is having. So whenever you are taking any drug, definitely it's not going to interact with only one target. It is going to interact with multiple targets because of the presence of the functional groups. For example, suppose if you are taking a drug A that is having only a single target, it's not going to interact with any other target, then it will be the therapeutic outcome particularly. But actually it will not happen because the drugs and the receptors are having different functional group. So whatever the drugs you are going to take, it is going to interact with multiple target. Either, for example, you can take 2-3 target. So any one of, suppose you are taking the first target, that is therapeutic outcome, then the other two will be responsible for the side effect. Or the second one will be the main effect, main therapeutic, or third one. That is depends upon the drug and the target what we are taking. So there are different pathways. So definitely one target will be the therapeutic outcome. The others will lead to the side effect. So this is the basic concept of the drug repurposing. So drug interact with several receptors. Just now I told you it is going to interact with different target. For example, you can take diazepam. So mainly it is going to bind, inhibit the benzodiazepine receptor, but it's also inhibit phosphodiesterase. Same thing when you are taking carbazine, it is main antagonist for kappa A, kappa alpha receptor, but it is also is a good inhibitor for monoamine oxidase, like that butyrylchlorine also. So drug repurposing or repositioning, because the people are used both the things interchangeably. But there is a minor changes there in drug repurposing and repositioning, but both are telling about the new use of old drug only. But in the methodology, there is a minor change, minor differences there. I will tell that the process of finding new use outside the scope of the original medical indication for existing drug is known as the drug repurposing. Means it is simply the new use of old drug. That only the title slide itself I also mentioned. So it is called as drug repurposing, drug redirecting, reprofiling, redosking, and rescuing, therapeutic switching, and repositioning. There are many names. So all it's indicating the new use of old drug. So drug repurposing concept, which was means come to in action in early 1990s, but actually it was followed from 1920 onwards. But actually it is accepted in 1990s only, means just 4 decades before. With the success of old drug in the treatment of several diseases, scientists are becoming more and more interested in drug repurposing and repositioning. So just now I told you there is a minor difference in drug repurposing and repositioning. Drug repurposing, it is like a serendipity, means opportunistic, means when we are doing something, we find out something else by accidentally or by luck. We are find out. For the best example for that one is sildenafil. When the scientists developing the drug for antihypertension and angina pectoris, accidentally they found out this is the good drug for the treatment of erectile dysfunction. That is one of the examples for drug repurposing. And repositioning, that is minor difference. It is a systematic approach. It is a drug repurposing is a serendipitous or opportunistic. But repositioning, it is a systematic research method. We are going to follow the systematic, means we have to use the computational method, experimental method to find out the new use of the old drug. That is repositioning. But both are used as interchangeably by the scientist. So that is the minor difference between these two. But actually, all are using either repurposing or repositioning. So new use of old drugs, all it's indicating new uses of old drugs. So what is a drug repurposing? It is simply a recycling, what we are doing in paper recycling and on the plastic recycling. Same thing here, we are going to go for recycling of medicine. That is the concept of drug repurposing. Means one medicine is suitable for me for treatment of one disease, but the same medicine will be suitable for the another person with another problem. So that is the concept of the drug repurposing. So what to bother us to go for drug repurposing? So already I mentioned few things. So anyway, we can see here what are the things why we have to go for drug repurposing. The first thing is the productivity in the pharma industry. As already we have seen, they are spending a lot of money, billion and billion dollars to develop the drugs. The failure rates are more, 80 to 90% failure were there in clinical trial, and particularly the toxicity problem. Then the number of drugs invention also, it's less. You see here from 2001 to 2020, this is the comparison table for new molecular entities and the drugs approved. And this is the blue color is indicating the top 16 companies, the top 16 pharma industries in the world, how many drugs they are producing, and other companies, what they are producing, the new molecule entities. So you can see the comparison, the ratio, and the drugs which is approved from this things in FDA approved drugs. So next one is pricing in rare disease. So this is the comparison chart, but it is somewhat old chart, 2012 to 2016. So you see the people, how much they was averagely spent per patient for particular disease. In 2012, you see 116, but in 2016, 140. So now we are in 2024. You think how much we are spending to treat a rare disease, that is neglected tropical disease, that is called as rare disease. There are 7,000 rare diseases are there now, which means it is affecting the small group of people. So the pharma industries or government, they are not concentrating more on that area. So that type of disease, we have to spend a lot to treat those type of disease. Only it is called as rare disease or neglected tropical disease. Yes, many of the non-orphan countries, non-orphan disease, you see non-orphan, how much they are spending, very less compared to the orphan disease. So the mainly the countries like USA and UK, German, all these countries, they are spending more for to develop the drug for the non-orphan diseases, like what you can tell, cancer, HIV, all these diseases. They are not concentrating on the non-orphan diseases. So that is the reason for why we ought to go for the drug repurposing. One is the productivity, then time consuming, then pricing the cost wise. Then the difference between traditional approach versus drug repurposing for drug discovery and development. So when we are talking about traditional approach, we have to start from the scratch. Scratch means you have to find out the disease, you have to find out the target, you have to optimize and validate the target, then you have to find out the hit molecule optimized to lead optimized to drug molecule. In that, then you have to synthesize the drugs, then you have to go for all the preclinical studies, clinical studies. Then after clinical studies, you have to get the approval. Then after approval, phase 3 trials, then the drug will come to the market. Then post-marketing surveillance will be there. So it will take 10 to 17 years to complete the conventional approach or traditional approach to drug development. But when you are talking about drug discovery and development, so already I told we are going to see the new uses of old drug, not only the old drug, even the drug which is not approved for any specific disease, but it has to pass the means. Already many of the drugs, it passed the phase 1 trial, but when it moved to phase 2, they failed in phase 2 trial. Means it is safety, but when it's coming to efficacy, that those compounds are not showing the expected efficacy against the particular target. So that drugs are failed. So we can take those drugs also, because already it is proved those drugs are safety. So the phase 1 trial already it's passed. So it's indicating the safety of the drug. But the drugs, those drugs are not showing the expected beneficial effect on the particular disease. So it is already passed the phase 1 clinical trial. So we can take those drugs also for drug repurposing. So no need to go for the phase 1 clinical trial. So we can reduce the cost and time what we are going to spend for the phase 1 trial. So don't think that the drug repurposing is only we have to take the old drug which is already approved by FDA. It's not like that. We can take the drug, the chemical compound which has already passed the phase 1 clinical trial but failed in phase 2 or 3 clinical trial. That also we can take. But we have to do one more extra step when we are taking phase 1 clinical trial passed drugs but failed in phase 2 clinical trial. But when you are taking the FDA approved drugs, no need to worry about the safety and safety. We can directly go for phase 2 and phase 3 trials. Then the optimization of the drugs is also not that much in when we are taking the FDA approved drug. So here, when we are talking about FDA approved, the drug repurposing, it will take maximum 10 to 13 years. 10 to 13 years. The first one is 10 to 17 years. Here, it will take maximum 12 years. The drug repurposing take maximum 12 years to bring the molecule into the market. Because here in drug repurposing, we are not going to design the molecule, we are not going to optimize the molecule, and we are not going to spend any time or any cost. We are not going to spend anything in the designing of molecule, the drug development. The initial stage near about 5 to 6 years we can save in that area, because we are going to take the molecule which is already approved by FDA, which is in the market, or which is the drugs which is failed in phase 2 clinical trial, we are going to take directly. So no need to worry about the designing of the molecule, synthesis of the molecule, the safety of the molecules. So whatever the years we are spending here, so this many years we can save, means 5 to 6 years we can save. So the time wise it will be better, and the cost wise also means only we are going to spend a lot for to synthesis and develop the drug. So here first one is compound identification. In drug repurposing, what you have to do, you have to find out the compound for the particular disease. That is the first step. The second step, compound acquisition. So compound acquisition means we are the drug safety approved drugs. So we have to check whether it is under patent or it's having IP rights, whether it is under the patent or it is the patent time is over, because many of the drugs they are following the US patent. So once they have got patent, the patent will be longer for 20 years. So we cannot use that drug for repurposing. We can use it, but we have to get the license from the concerned company. If the same company, those who have introduced the drugs, they are doing, then there is no issue. They will get the extension of the patent for another 3 to 7 years. Or we have to use the patent expired drugs, which is not under the patent. We can use that drug for drug repurposing. That is the major drawback in drug repurposing, because many of the drugs under the patent, we cannot use it without the proper license. So the intellectual property is playing a major role in drug repurposing. Then you have to develop the drugs. Develop the drugs means the development, the clinical trials, and you have to go for registration and post-marketing studies. So that is the difference between the traditional approach and drug repurposing. This is also one of the reasons why we have to move towards drug repurposing. Because when you want to synthesize a new compound, already I told, if you are synthesizing thousands of compound, sometimes one compound may be so the potency against the particular disease, or sometimes none. So the failure rate will be more in the traditional drug discovery purpose. So other things in traditional drug development and drug repositioning, the average cost to bring new drug to market, that is $1.24 billion. This one is less cost, because we are going to reduce near about 30 to 40% of this cost. Then time record. So just now we have seen 10 to 17 years for traditional and 3 to 12 years for drug repositioning or repurposing. So here we are saving 5 to 6 years. Here we are saving near about 30 to 40% of the cost. Then failure rates. Here it is high. It is 80 to 90% failure rates are there. According to the literature, about 80 to 90% failures, particularly due to its toxicity. But here the failure rate is 50 to 60%. According to the literature, it is 50 to 60% in drug repositioning. Then increasing focus on drugs to treat chronic and complex disease. But here our focus, the drug repurposing, the focus should be for rapidly emerging and re-emerging infectious disease, particularly the orphan disease. That is the main thing. Because the orphan disease, the companies or the government, they are not ready to spend. Because it is an orphan disease, it is a rare disease. To spend this much million to develop the drug for the rare disease, there is no meaning. So only the tropical diseases are neglected. Near about 7,000 neglected tropical diseases are there. So for that, what you have to do, you have to find out the use of the old drug to treat the rare disease. That is the main concept for drug repurposing. Because here we are not going to spend much, either it is the cost wise or time manner, or the failure rate also will be less. So this is most suitable to find out the use for the rare disease. So drug repurposing versus traditional drug development, the challenges in clinical trials and intellectual property issues. That is the major challenges in the drug repurposing. Then drug candidate for repurposing. Which of the drugs we can take it for repurposing? As I already said, either it should be a FDA approved drug or the drugs which is failed in phase 2 clinical trials. So discontinued for commercial reason, because that is already approved drug, it was in the market, but due to some reasons there is no commercial value, so it is withdrawn from the or discontinued from the market. So those drugs we can take. Then patent close to expiry, generic available. So which is near to expiry. So it is going to expire means we can use it for drug repurposing. We can get the license, means the patent for 3 to 7 years. But the new medical entity, we can get license for 20 years. So the patent timing for repurposing drug is 3 to 7 years only. So many of the companies, they are not ready to go for drug repurposing. This is one of the reasons, because they are going to get the patent for 3 to 7 years only. So they are spending a lot and they are not going to get back their monetary benefit. So many of the companies, they are not ready to go for drug repurposing. That is the reason. Then current branded compounds, these are available on the market but remain protected by intellectual property. So that only I told. So you have to get the license, means permission from them, then we can go for those drugs. Then fail to demonstrate efficacy during phase 2 and phase 3, no major safety concerns. That only I told. The safety concern we are going to check in phase 1 trial. So already that the chemical compounds are passed through phase 1, but it is not, it's failed in the phase 2 or phase 3 trials. So those type also we can take it, because there is no major safety concern, because already it's passed the phase 1 clinical trial. So those type of compounds also will be the suitable or apt candidate for drug repurposing. Then working what we know. So we have to collect the information. From that, we can go for the things. So no de novo drug discovery. De novo means from the beginning, from the scratch, we are going to synthesize the drug. De novo means from the beginning. So there is no beginning drugs. That is no de novo drug discovery. Then fast, cheap, good for rare disease. Just now I mentioned why it is good for the rare diseases. Then known safety profile and side effect. That is the other things what we come to know from the literatures. And then history of human use. That also we are having for the drugs. Then reduced requirement for early stage clinical trials. Then known pathways of action. Then ideas or evidence for repurposing candidate. So these are the things what we know. We can start with these things. So repurposing of generics should be appealing to the wealth of data available on their use in humans. Because when we are taking the FDA approved drug, we are having all the data, but it should be available. Then you can use that available data. So half of the process is complete there only. So remaining half only we are going to do. Then, however, whatever it is, then hard to secure the intellectual property rights on generics. That is the major, already I told, it is the major issue in drug repurposing. Why the companies are not concentrating on drug repurposing? This is the main reason, because they are not getting the intellectual property or patent drugs. So it is very difficult, and it is a complex and time-consuming process. So you have to get the license from the real owner of that patent. Then you have to apply for the license. It is a complex process and it is a time-consuming process again. Then mode of use patents are hard to defend. That is another thing. Then the major thing is off-label prescription of alternative generic is hard to detect and to prevent. The best example recent the pandemic situation, when the COVID-19 was identified, immediately they have used the computational technology like reverse means docking, reverse docking, many computational technology, they find out some drugs which is already available in the market is best choice for the treatment of COVID-19. For example, remdesivir, dexamethasone, and ivermectin. So these are the drugs they found out. That is called as the drug repurposing by using the computational approach. But none of the company is ready to produce the different means the formulation means the generic drugs. Yes, the doctors are having rights to prescribe the drug off label. Okay, that is the main thing. We cannot prevent that off-label prescription. Suppose for aspirin, it is aspirin is meant for analgesic and anti-inflammatory and antipyretic. But later on, it was invented to treat for it is a good anti-clotting factor, means antiplatelet. It will prevent the clotting. So we can use the generic because the doctor will prescribe the generic form. So when we are going, the company will go for the formulation of that. Then they have to go for some new formulation. Then they have to go for all the studies. Or they have to go for the phase 2, phase 1 also they have to do when they are changing the formulation. Then they have to go for phase 1 also. The safety also they have to check. So again, the process will be like same like the traditional approach other than the drug development. So that is the another problem. So it's very difficult to prevent the off-label prescription. Means the generic drugs will be prescribed for the other purpose other than it is intended. So here's a Food and Drug Administration section 505 with respect to approval of drugs. So 505B1 is the FDA approval for de novo drug development process, the new drug development process. 505J gives the pathway for generic to get approval by submitting the abbreviated new drug application. But this one is the main, that is section 505B2 gives the pathway for repurposed to get approval and allows the sponsor to rely on part of existing safety and efficacy data from previous trial conducted for the original indication of the drug for which the drug is already approved. So this is the FDA section which is approving the repurposed drugs. Then drugs approved under section 505B2 can obtain a minimum of 3 years to maximum of 7 years of marketing exclusivity. That is the another things why the pharma industries are not moving towards a drug repurposing. If it is a new entity, then they are having the patent for 20 years. But here they will get minimum 3, maximum 7 years. So they are spending a lot. They want to retake the money. Then it's very difficult within this time period, 3 to 7 years, according to the pharma industry is concerned. So repurposing for pharma, the repurposed drugs, how they are going to do the repurposed drugs. So we are going to use the same drugs, but we are going to change the delivery. Which delivery? The different delivery systems. Already you know whether it is oral or parenteral or nasal, whatever the drugs, the delivery system, or whether it is a controlled form or conventional form, either liposome, nanoparticle or microparticles or any other formulation, tablet, capsule, whatever it is, the delivery things. Sorry, that is the delivery is towards which the formulation. Just now I mentioned which formulation, either it is a liquid formulation or solid formulation, whether it is tablet form, capsule form, other things, and dose. That is the most important things. For example, paracetamol, we are taking, for example, aspirin. So this is the particular dose is for analgesic purpose. When we are going to use as antiplatelet, there is a different dose. When you are increasing the dose, automatically it will increase the side effect also. So you have to be very careful when you are concerning about the dose, formulation, and delivery system also. When you are doing these things, when you are going to repurpose the drugs, so we are going to play either one of these, either dose we have to increase or decrease. The formulation, we can go for different formulation, or the delivery system, different delivery system we are going to use. So when we are doing this, definitely you have to go for the safety wise, because already the safety wise they have we don't know what they have developed for. So the safety wise also means phase 1 also will be included. So again, IP intellectual property, but requires new safety data for human use. That only I told. When you are doing any changes in the dose or changing the formulation or changing the delivery system, then you have to go for safety data again, even though it is available already, it passed the phase 1 clinical trial. But when you are do changes in any one of these properties, then you have to get the safety data again. So repurposing without reformulation work can appeal to academia. So drug repurposing profile. So drug, already I told, it's going to interact with known target. So known therapeutic indication. This is drug, a known target, it is known use. But the same drug interact with this target, but it will produce the new therapeutic indication. That is first purpose. The second one is drug, known target, known therapeutic indication. The same drug will interact with the new target and new therapeutic indication. These are the two repurposing profile. What when we are talking about drug repurposing, there are two profiles. One is the drug acting on the old target, but it will give new therapeutic indication. The next one is that old drug, but it is going to interact with the new target and new therapeutic indication. So drug repurposing profile, the known target, new indication. For example, we can take finasteride, that is known target, that is old indication is benign prostatic hyperplasia, but later it is male pattern baldness. It is used to male pattern baldness. Known compound, new target. For the best example is thalidomide. I think you know the tragedy of thalidomide. Earlier it was used for morning sickness for the pregnant ladies. When it was given to the pregnant ladies, it leads to teratogenic effect in the malformation of the infants, the child. So it is withdrawn from the market. But now again, the FDA is approved this thalidomide for to treat leprosy and the multiple myeloma, the blood cancer. Yes, it is a good choice for that drug, but the indication should be mentioned when we are going for repurposing, the dose difference and other things. Along with that, the indication will be there. Thalidomide should not be prescribed for the pregnant lady. So with that indication, nowadays it is approved for to treat leprosy and myeloma. That is known compound, new target, because the thalidomide is going to interact with some new target and responsible for the new therapeutic effect. Then drug repurposing, there are profile, there are two things. One is serendipity, the another one is hypothetic drawn. The serendipity, it is by luck. When you are searching for something, but you got it something different. The same thing also happen in drug discovery. They are trying to find out a drug for some disease A, but later they found out that is suitable, the drug is suitable for the treatment of disease B. So that is the serendipity. It is by luck or by opportunities or by accidental. You can take any meaning. So the best example is sildenafil. So it is an inhibitor of CGMP, specifically phosphodiesterase 5 inhibitors, and unsuccessful for hypertension angina. But the drug was initially invented to treat hypertension and angina, but later they found out it is successful for male impotence and pulmonary artery hypertension, means to treat erectile dysfunction and treat pulmonary artery hypertension. And recently they found out it is the one of the drugs used to alleviate of altitude sickness and jet lag. For that purpose also, nowadays the sildenafil is used. That is serendipity. Then drug repurposing. So virtual or physical library of drugs. When we are using physical library of drug, then you have to go for high throughput screening. When you are using virtual drug means just drawn and stored in your computer. That is, we are going to find out by virtual screening. So you can use any one of the drugs. If you are using virtual screening, then you have to take the drug directly for the activities. If it is physical library of drugs, means the drugs are already available. So you have to use any experimental technique, that is biomolecular screening. So you can find out the new drug activity, then confirmation of clinical effect, then go for phase 2 and phase 3 clinical trials. Then we will get the repurposed drugs. So this is the process of how they are doing the repurposing, either it is a virtual screening of drugs or physical library of drugs. So this is the roadmap for drug repurposing. So we can use any one of the techniques to find out the new drug. So either it is phenotypic screening, in silico screening, drug omics data, side effect similarity. So a lot of things are there. So by screening about, we have to get the information about these things. Then we can find out the new target, world therapeutic indication. This is the drug. Either it is new target, world therapeutic, new drug target, new therapeutic, old target, new therapeutic indication. Anyone is possible. Then go for finding evidence in data. So means you have to do the experimental and find out the data, then go for in silico screening evidence, then generation of proof of the concept, then general experimental evidence, then move to clinical trials, then further FDA approval and marketing, the successful market drug. So methodologies used for drug repurposing. Drug repurposing demands an extensive literature research recording the drug profile and targeted disease ultra mechanism that only just now we have seen. You have to collect the information from the different databases. And latest reliable data pertaining to the development and progression of the disease. You should know the full development progression of the disease, specific biomarkers associated with different stages of the disease, because each and every stage there are different markers will be there. So you should familiar with all these biomarkers. Then any research focusing on genetic disorders, then selection of suitable approach for repurposing the drugs. There are different approaches out there. So you have to select which approaches is apt for your drug. So you have to select that one. So this is the hypothetic driven approaches, signature-based strategies, target-based strategies, drug-based strategies, and literature-based strategies. So these are the different strategies. This is one is mainly based upon the gene structure, that is signature-based strategies, then target. So whatever the target, based upon the target, you have to select the drugs. Then drug-based strategies. It is based upon the drug molecule. We have to find out the target. Yes, we know the drug molecules, the main therapeutic effect and side effect. Then when we are knowing the side effect, then we can take two drugs which is having the same side effect, means it is going to act on the same target. So it will means what you can tell, support each other, means it is going to give the same effect. So there we can expect the synergetic effect, because both the drugs are having same side effect, means it's going to act on the same receptor. So we can expect the synergetic effect. So we can go for combination therapy. Then we can collect the information from literature. Then we can go for the drug repurposing. So signature-based strategies, identification of genetic factors like gene expression, gene regulatory, and transcription factors involved in the pathogenesis of the disease. So computational map tools like connectivity map is a web-based tool that established similarities among the drugs based on genetic expression profile. So we can use this one. So this is the another developed database which consists of reported single nucleotide and their associated generic trait expression. So we can use these things for signature-based strategies. So how to do? So particularly by using the omics technology, that is repurposing ideas. So omics, understanding all of the genes or proteins in an individual, is at forefront of science and can be used to find repurposing opportunities. For example, this is the gene expression level of different genes in a disease affected cell. So we can take the genes in the disease affected cell, then find out the expression. So for example, this is the expression. See, some of the genes are overexpressed and some of the things are underexpressed. So this is the condition in disease. So it is called as a disease signature. Then what we have to do? Then repurposing ideas. So this is, we know this is the disease signature. So we have to find out the drug which is having this signature, drug signature. So any drug showing this signature, then this will be the suitable or apt drug for this particular disease. So what you have to do? So this is disease signature and drug signature. So you can match these things. Then we have to find out the therapeutic result. So this will be minimized. So everything will become normal. So here your gene is overexpressed, but your drug is underexpressed. Here underexpressed, your drug is overexpressed. So the effect will be nullified, means minimized by each other. So we will get the normal value like this. We will get the normal effect. So this is called as therapeutic effect. So this way we can find out the drug for the disease by using the signature-based approach. So this is one of the study was reported for the identification and preparation of drug for high-grade serious ovarian cancer. They have taken near about 7,000 drug from 31 classes. Total drug is 7,000. It is belongs to 31 classes. They have done the studies. Then they found out there is the particularly the NSAIDs are near about 25%, showing the effect on this particular cancer. In that, they found out the trexalicacib and nimucilide. They are those who are selective COX inhibitors are showing better response against the cancer, and non-selective inhibitor indomethacin shown better the top compound in the treatment of ovarian cancer. So target-based strategies involve the study of particular protein or biomarker involved in the pathophysiology. So yeah, dysfunctional protein or mutated protein or a misfolded protein may lead to pathological crosstalks triggering the disease development. Means based upon the target, we can develop the strategies, whether it is a protein or any other biomarker, whether it is mutated or misfolded. Based upon that, we have to develop the drug. Then target-based strategies also, there is a directionality map establishes the effect of drug on gene via protein interaction in computational approach. Then we can use the well-known technique, that is docking techniques or molecular dynamics, to find out the target and receptor interaction. For example, here they have used inverse virtual docking, that is a reverse virtual docking. They have used first, they have done the studies for set up R1 compounds designed for JMJD3. So they have used in vitro reports, but it's showing poor efficiency against the JMJD3. But the same drugs they have used in inverse means reverse virtual docking, then against set of anti-cancer protein, other anti-cancer protein, then they found out these drugs are against means discovery of ERB4 inhibitory potential. These drugs are having this potential, that is gastric carcinoma. So inverse virtual docking of orphan compounds of sulfonamide class. This is sulfonamide compounds. So that is best choice for to treat the gastric carcinoma. That is one of the target-based technique. So molecular dynamics, the other things, the simulated techniques, which is also used in the target-based strategies. So this is the process how to do FDA-approved drugs. So you have to do docking against wild BCR-ABL1 and TRK-resistant mutants. Then from that TRK with comparable docking score of, that is ponatinib standard, were subjected to molecular dynamics. Means first you have to do docking studies against the wild variants and the TRK-resistant mutant proteins. Then you have to find out which one is suitable compound. Then that is, they have found out ponatinib is standard drug, which is showing effect on these two. Then it was subjected to molecular dynamics. Then find out these drugs. So drug-based strategies, already I told, one drug is going to affect on the multiple target, that is polypharmacology. It is called as polypharmacology. So gaining importance due to huge availability of the computational approaches developed, which exploring the hidden mechanism of drugs. So there is a lot of techniques, QSAR, pharmacophore mapping or modeling, then drug similarity approach. Many techniques are there. I don't want to go into that. So we can use any one of the techniques, then we can find out the drug is suitable for which target. Then pharmacophore modeling. So pharmacophore denotes the significant interaction features of ligand responsible for agonist or antagonist of biological response. Then repurposing based on clinical side effect. So we can use the web-based tools, that is CITR and clinicaltrials.covet. So by using this, we can find out the side effect of the drugs. Then we have to develop that side effect as the main effect. So the approach based on the drugs with similar side effect exert similar mechanism. So what we can expect, we can combine the two drugs which are having the similar side effect. So we can expect the synergetic effect of that. So that will be used for drug repurposing. So literature-based strategies. So as you all aware that we can collect the information from the literature, then we can use it for drug repurposing. Particularly, we can use the medical subject heading, which is available in the PubMed terms to retrieve relevant information. So this is the process, how to do the literature survey process. This is one of the example. So then computational and experimental approaches used in repurposing. Just now we have seen all the techniques. So it is all the techniques I have compiled here. That is I have taken from the Puspakam et al. From Nature Review. Then the databases, because we need the information, either it is a protein or a drug, we need all the information. So there are different databases used in drug repurposing. So for target, we can use any one of this database. Then pathway mix data, we can use any one of this database. Then drug, if you want to get information about drug, you can use any one of this database. Then toxicity data, go for this database. Genomic data and clinical trial and adverse effect data. So there are many databases maintained and many of the things are freely available. So we can collect the information from the respective data for what we need. Suppose we need toxicity data, then you can go for the database Tox21 or Toxcast. If drug, then you can go for DrugPang or PubChem or DrugMap Central. We can get the information about the drug, like what are the physical properties, what are the other things, what is the molecular formula, solubility, all these things we can get from this database. Then compound libraries, the National Institute of Health Clinical Collection, we can get the libraries, we can get the information from here, or National Institute of Neurological Disorders and Stroke, Trustwick libraries, chemical libraries, the Clinical and Translational Science Award Pharmaceutical Asset Portal. So these are the some libraries where we can collect the information. Then the main drawback and another drawback in our drug repurposing is we cannot use the pro drugs because it is problematic in repurposing. So we know the pro drug, the drugs which is not actually, it's not having any pharmacological activity, but once it will reach into our biological system, intestine or stomach, there it will be hydrolyzed or released from the drug, and the drug, active drug, will be released from the product. Then it's having the pharmacological activity by action of different enzymes. So when it is acted by different enzyme, then it will change to different molecules. So we cannot use this products for drug repurposing because the products are not biologically active, but once it is hydrolyzed or released from the product, then only it will be active form. So we cannot use this one as your, the products as for drug repurposing. Then other structural complexities, tautomers. Suppose the compounds is available in tautomers, then epimerization or covalent or reactive, active metabolites, hydrogen, elimination of stereoisomers. So these type of difficulties are there. We have to consider when we are going for drug repurposing. These are the structural complexities we have to face when we are talking about the drug repurposing, including prodrug, the other structural difficulties what we are going to face when we are doing drug repurposing. For example, you can take metazolam. It is at neutral pH 7.4. It is available in this form. But when it is in low pH, then it will be, the ring is opened. It is an entirely different molecule. But in higher pH, it is 1-hydroxy metazolam. That is 10% of the dose converted into this form. Because this CH3 will be converted into CH2OH. So this is the problem when we are talking about the stereoisomers. Then novel screening platform, then screening of compounds using in vitro and in vivo system, high throughput screening, that is the recent technique which is used in vitro binding assays. Then we can go for cell-based assays and small organism-based screening system. These are the novel screening platform where we have to use in drug repurposing. So these are the some methodology that drug to repurpose. So drug target way, pathway, disease oriented, and side effect oriented. This is the method. Just now we have seen we can use the drug molecules like that, or we can go for target based, pathway based, or disease based, or side effect based. So these are the methods. Which method they have used? For example, this one they have used molecular talking pharmacophore modeling. Then this is the resource they have used and reposition drugs. Example, This one, just we have seen in the first slide itself, bupropion and thalidomide. Like that, you can see all of the drugs which is used, means repositioned drugs. So these are some list of repurposed drugs for different. This is the original indication. For example, drug allopurinol. First, it was invented to treat cancer, but later they found out it is the best drug to treat gout. So like that, amantadine, that is mainly antiviral drugs, but later on it is a after drug to treat Parkinson disease. So like that, there are many. I think recently I've seen one of the article which was published in the later quarter of 2023, published by Golani et al. From India. They have compiled near about 125 drugs. Yes, nowadays, recent years, it's not earlier, the recent years, whatever the drugs approved by FDA, in that 30% of the drugs are repurposed drugs. Means 3 drugs are approved by FDA, in that 1 drug is repurposed drug. That is the information what I have collected from the literature. But it is in recent years, not in the earlier years, but not in the overall, but recent years, that 1 in 3 is the repurposed drug. So these are the some drugs which is used for pulmonary arterial hypertension. Then all these tables, what I have shown, all are repurposed drug for the particular system wise. This is the drug repurposed for central nervous system. Then this is for CVS. I have taken all this table from this reference, that is Mira et al., which is published in Journal of Physiology, Pharmacy and Pharmacology 2022. So these are the drugs in different system. So these are the drugs, antimicrobial drugs, repurposed drug in endocrinology, and repurposed drugs in cancer chemotherapy, and repurposed for miscellaneous indications. The indications are given here. Then silent examples of repurposed drugs in pipeline, means already it is in phase 2 and phase 3 clinical trials. And examples of blockbuster repurposed drugs. Earlier we have seen one of the top blockbuster drug is seltenafil, then samsitafin, relaxifin, rituximab. All these drugs, trimethyl fumarate, all these drugs are the blockbuster drugs, which is invented by drug repurposing. Then example of repurposed drugs approved by FDA in 2019. So these are the drugs which is approved by FDA in 2019. Then drug repurposing in India. So India is also, government is also supporting, for some of the companies are supporting for doing repurposing, particularly for tuberculosis, HIV, malaria, and neglected tropical disease like leprosy, lymphatic filariasis, and visceral leishmaniasis, and some lifestyle disease, diabetes, hypertension, and IHT cancers. Then many of the companies, already I told, they are reluctant to invest in R&D of drugs for infectious disease and neglected tropical disease. Already I told the reason why they are reluctant to invest in the R&D for these things. So drug repurposing in India, the global initiative based on public-private partnership models. So these are the things. Then the drugs paramomycin and miltefosine repurposed for visceral leishmaniasis following trials in India. So these are the 2 drugs where they did trial in India. Then companies dedicated to repurposing, that is Conaxis, which is based on Bangalore, then Jubilant Biosys and GVK Bio, Hyderabad. These are some companies, those who are doing the drug repurposing. And challenges in drug repurposing process, optimization, inclusion and exclusion criteria and selection of target population. Then incorrect subject selection lead to disaster effect of the drug instead of expected one. So that is the major drawback, not only in drug repurposing, in new medical entities also, it is a major problem. The best example is thalidomide. Then achievement within timeline, dosing regimen, route of administration, and optimizing the formulation without stabilizing the drug is the another major issue in drug repurposing. Then repurposing of drug aims different group of patients with a different set of physiological condition. Therefore, unexpected adverse events may be anticipated. Then repurposing, drug combination demand, prerequisite teton, drug interaction, pharmacodynamics and kinetics of the drugs. When we are going for the combination of the drugs, just now I told when the two drugs are showing the same side effect, means it's going to act on the same receptor. So we can use these two drugs as a combination to increase or synergetic, to expect the synergetic effect. But when you are going for combination, you have to study about this drug interaction, whether these two drugs are interact by themselves. Then the pharmacodynamic and kinetic properties of these drugs, whether one drug is going to influence the other drugs pharmacodynamics or kinetic properties or not. So all this information you have to collect. That is another challenge in drug repurposing. Then need of new phase 1 trials, that when the compounds are we are changing the dose or formulation or delivery, then you have to go for again phase 1 trials. Then if your solved compounds is brought back to the clinical trial, it has to pass through all the regulatory requirements, like filing from the NDA to the marketing. Then IP related issues, already I told what are the IP related issues. Then low economic returns. That is the major reason why the many of the pharma industries are not ready to invest in drug repurposing, because the patent is value for 3 to 7 years only. The future direction and recommendation, new method of use patent for an old generic drug should be utilized. So we have to do some changes in the patent. Then limit off-label use of generic drug by developing unique formulation or dosage. So that is the another things recommendation we have to, because we cannot prevent the off-label prescription, but we want to prevent that, we can go for new formulation or dosage. But when you are going for new formulation and dosage, then you have to check the safety level. Then develop financial incentive to encourage generic manufacturer to participate in drug repurposing, such as extended market exclusivity, royalty arrangement. So you have to do something to encourage the generic manufacturers. Then collaboration between, this is the most important thing, the larger pharmaceutical companies and academia and small companies. So you have to go for collaborative work. That when in collaboration only we can achieve many things. Means in academic you are not having the industrial the structures. So we can collaborate with industry, small industry or larger industry. Then we can develop the track. Means we can do the repurposing. Then funding opportunities specifically for repurposed drugs in collaboration with industry. That is the another direction. Then collaborators between patent advocacy groups, philanthropic organization and academia, particularly to explore generic marketed drug for rare disease. Small but robust proof of concept studies funded by non-profit organization might provide enough information for a physician for off-label use without regulatory approval. Then whatever it is, despite of the hiccups during the process, the importance of the drug repurposing using in silico approaches has gained importance in the drug discovery field in present pharma industries. The best example is the recent pandemic COVID-19. So that is the best example for the in silico, what is the role of in silico in the drug repurposing. So repurposing is the future. Traditional drug discovery routes cannot deliver for all rare diseases on their own. Yes, already I told the traditional drug discovery, we are going to spend a lot of time. We are going to spend cost-wise, it is not economic. So we are spending that much for rare diseases, then it is meaningless. So you have to find out that all the drug to be used for the rare disease. So repurposing offer a quick, cheaper and collaborative route to the development of effective treatments. Academic and patient-led collaboration are proving successful and more approaches are being developed right now. So sponsoring for drug repurposing, there are many sponsors. They are sponsoring for the drug repurposing purpose. So like National Center for Advancing Translational Science, MRC AstraZeneca, World Health Organization and other public-private sectors are sponsoring the conceptual proposal on drug repurposing with huge expectation towards solution for unanswered issues in existing medical condition and therapy failures. So these are the references what I have used, where I have collected the information. And particularly I've used some PPT here. Also, I have taken the slide from these people. I'm thankful to all these contributors. So these are the further reading. If you're having time, you can refer these two articles. They are giving a lot of information about this truck repositioning or truck repurposing. And recently, just now I told, I have read a paper from Golani et al. In, I think, Russian Journal of Bioorganic Chemistry. So that is, it is a well-compiled review article. So you can go through that article also. They have given here about 124 repurposed drugs. They have compiled the data. So whatever it is, the poor research can end with universally recognized medicine, and conversely, a brilliant rationally can remain sterile. That is the common concept for whether it is traditional drug design or drug repurposing. So with that, I thank all the participants for your patience and listening the talk. So I can hand out to the convener.
Thank you so much, Dr. Ravichandran, for
Thank you,
delivering
sir.
a marvelous session on a very novel topic, drug repurposing, exploring new therapeutic avenues for existing drugs. You started the session by the quotation saying, discovery of new drugs starts with an old drug.
Yeah.
You also explained the drug repurposing as a new use of old drug or selective optimization of side activities. You also told the difference between drug repurposing and repositioning. You discussed the traditional approaches versus drug repurposing for drug discovery and development. You also told that the cost, time, and failure rate is low in case of drug repurposing in comparison to typical drug development. You also discussed that the patent granted for drug developed through this technique is 3 to 7 years. But this technique of drug development is fast, cheap, and good for rare diseases. You also shown us the route map of drug repurposing. You've shown us a lot of examples of drugs that are repurposed. Thank you so much, sir, for educating all of us on the latest approach of drug discovery, that is drug repurposing. Now, there were
Thank you,
some
sir.
questions from the participants, sir, but I think
Yes, sir.
you have covered everything in your presentation. All
Thank you,
of the
sir.
questions are answered very well, and all of the questions answers are covered in the presentation only. But still, I will be asking one or two questions from the participants, sir.
Yes, sir.
So, how do personalized medicine approaches influence drug repurposing efforts?
Yeah, personalized medicine is when you are going to do the first, you have to know the gene profiling of individuals. So that time it will be more useful. But when we are going for personalized medicine, that is entirely different concept from drug repurposing. Yes, each and every individuals, we are having different gene setup and response. But that time you have to think about repurposing. So it will not be useful that time, definitely. So personalized medicine means you have to, based upon the genetic, you have to design the drugs. Means you have to design the, confirm the dose and what is the route, everything based upon the individuals.
Right, sir. Sir, the next one is, can you elaborate on the process of clinical trial design for evaluating repurposed drugs?
Clinical trials, the same thing only, same
Right.
clinical trial, there is no difference. The same phase 2, phase 3, phase 4,
It
everything
will be
is
same,
same.
like the
Yeah,
traditional
same one.
drugs.
Yeah, same only. We have some of the drugs we
Right,
have to
sir.
go for phase 1 when we are changing. Just now I told, when we are doing some changes in the dose or formulation or delivery system, then you have to include phase 1 trial also. There is no change in that. The same
Right,
common
sir.
traditional drugs, what we are using, the same clinical trials we are going to use.
Right, sir. So, rest all the question are answered, already answered by you in the presentation. So here we will come to the conclusion of the webinar. So in conclusion, our 48th SPSR International Webinar on Drug Repurposing, Exploring New Therapeutic Avenues for Existing Drug has been stimulating and enlightening session that underscores the immense potential of repurposing existing drugs for novel therapeutics purposes. Throughout this webinar, we delved into the innovative strategies, cutting-edge research, and promising outcomes in drug repurposing, highlighting its significance in addressing unmet medical needs. accelerating drug development timelines, and reducing costs associated with traditional drug discovery. We have witnessed compelling presentations and insightful discussion from esteemed speakers, shedding light on various aspects of drug repurposing, including computational methodologies, clinical trial design, regulatory considerations, and case studies exemplifying successful repurposing efforts. The diverse perspectives shared have emphasized the interdisciplinary nature of drug repurposing, involving collaborations across academia, industry, regulatory bodies, and healthcare practitioners. Moreover, the webinar has underscored the importance of leveraging existing pharmacological knowledge, high-throughput screening technologies, and data-driven approaches to identify new therapeutic indications for approved drugs, thereby maximizing their clinical utility and benefiting patients worldwide. By repurposing existing drugs, we not only expedite the delivery of safe and effective treatment, but also contribute to sustainability in healthcare by optimizing resource utilization and reducing drug development costs. As we conclude this webinar, let us remain inspired by the potential of drug repurposing to transform the landscape of medicine, offering hope for addressing complex disease and improving patient outcomes. On behalf of SPSR, we extend our gratitude to the speaker, participants, and organizers for their invaluable contributions and engagement, and we look forward to continued collaboration and innovation in advancing the field of drug repurposing for betterment of global healthcare. We extend our sincere gratitude to Dr. Shashi Alok, the National President of SPSR, for gracing us with his esteemed presence today. In acknowledging your dedication and leadership, we express our heartfelt thanks for your invaluable contributions. In extending our deepest gratitude, we express our sincere appreciation to the esteemed speaker, Professor Dr. V. Ravichandran, whose generosity in sharing his profound expertise and invaluable perspective has enriched our understanding of drug repurposing, exploring new therapeutic avenues for existing drugs. A special acknowledgement is extended to our convener, Mrs. Monika Sabharwal ma'am, whose pivotal role in orchestrating this insightful event has been instrumental. We extend our heartfelt thanks to all the participants, recognizing the collective efforts that has effectively pushed the boundaries of knowledge and fostered a spirit of collaborative learning. Together, your contributions have made this webinar a resounding success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. Thank you for being an integral part of the 48th SPSR International Webinar. Your active participation and contributions have been invaluable, and we look forward to future endeavors and that contribute to the continuous betterment of healthcare and well-being for all. Thank you once again for your participation and unwavering commitment to the advancing the frontiers of science and medicine. As we conclude this webinar, let us remain inspired by the potential of drug repurposing to transform the landscape of medicine, offering hope for addressing complex diseases and improving patient outcomes. Have a wonderful day ahead. Thank you so much, sir.
Thank you, sir. Thank you. Thank you, one and all.